After the assay model developed, and we will use positive control and negative control to run dose response curve and to set up the detection limits, and also we'll set up the hit ratio, and we validate the model until we have a good signal noise sensitivity and can be run at the different conditions and for the different type of fraction and extract library. After the assay developed and validated, we are using integrated robotics technology to screen the library at a high throughput mode. And we designed the plate and running the sample in triplicate, and we can carry on thousands of samples screening every day and complete the screening in several months and the timeline. After we developed the model and validated the model, we put all our extract libraries and to screen it, and you have a distribution population of the hit, and most of the case you will see 0.5% or 1% of a total hit rate. And those hit compounds will be taken out to run dose response curve and to validate the hit, and then each hit extract will be fractionated into 88 fractions. Those fractions will be carried on the further screening on the same model until we find the positive fractions in those material. After screening the high throughput fraction library and we found active fractions, we are using different technologies to dereplicate those active fractions. One of the technology we are using is a high throughput TLC, and which can spot all the fractions on the TLC plate and develop it to look into how many components in there. And also we can use HPLC, MS, and HPLC, ELSD, or other detectors to analyze those extracts to look into the peak intensity against the biological functional intensity to identify which components in there are responsible for the activities. After we screen the high throughput fractionation library, we find the active fractions. We will use high performance thin layer liquid chromatography to detect, analyze those active fractions, and to look into how many components in those fractions. We also use LCPDA, LCMS, and NMR technology to dereplicate those active HTP fractions. We analyze the peak intensity from the chromatogram against the biological profile and pinpoint which peak correlated to the biological function. And we also use a bioinformatic database, such as a dictionary of natural product, which contain more than 200,000 structures and many different kind of plant marine species and names. And we use those information against our data coming from the dereplication process so that we can pull the structure information out of those active fractions. One of the example of dereplication we analyzed more than 20 fractions, and from green tea extract against the COX-1, COX-2 inhibition, we identified more than 10 structures from those fractions which corresponding to the COX inhibition activities, as you can see from this slide. After dereplication, when we decide to isolate the active components in large quantity, and we go back to bulk material to using 200 or more grams of biomass to restart the extraction fractionation process. Since we already pinpoint which peak we want to isolate in the dereplication process, we can carry on the large-scale isolation, fractionation, and the purification efficiently. We can finish the compound isolation in a couple of week time period. And after we isolate the purified active components, those compounds will put back to the screening model to retest, make sure the activity is still there, and also we will measuring the EC50 or IC50, and based on the dose response curve to confirm those compounds are active components. And after we finish the testing of active components on the primary screening model, we will develop the secondary model on different cell line or different system, and to confirm the functions of the active components and on the secondary or tertiary assay system. As you can see, coming out of this discovery process, we started with more than 5,000 or even more extract in the beginning of the screening. We further screen hundreds of thousands of fractions and isolate several hundreds of pure compounds, and half of them are active on the original screening model. And then we screen further in the in vitro and in vivo setting, and we eliminate those compounds without activity or weak on the potency. And by the end of it, we may have one or two product leads goes to the human clinical trial and eventually bring to the consumer product. Through the PhytoLogix discovery process, novel compounds will be isolated, developed. We also will find a novel usage of known components. And we are very actively pursuing intellectual property. We file hundreds of patents worldwide and domestically. And we have many issued patents, and including the licensed patents from university for exclusive usage. And we protect our core product and surrounded by intellectual properties and trademarks and know-how. And also we strongly, vigorously defend our IP positions. We know how important it is to our partner. In overall, Unigen work with plant collectors around the different countries, and also we have a very professional scientists and marketing teams who dedicate all their efforts to bring the best nature to mankind.